Although the rise in incidence of childhood obesity appears to have plateaued in some developed nations, the condition is still estimated to affect 124 million children worldwide, or an estimated 1 in 3 children in the US. Children who are overweight or obese are more likely to become obese adults and have an increased risk of poorer health outcomes in later life, including diabetes, heart disease, cancer, and overall mortality. Like obesity in adults, childhood obesity has been linked to impairments in executive functioning, although studies produce conflicting results. Executive function is an umbrella term for several different cognitive dimensions, including inhibitory control, decision-making, working memory, and reward sensitivity, broadly referring to a set of processes that enable planning, problem-solving, flexible reasoning, and regulation of behaviors and emotions. Children who are overweight or obese generally score lower on various measures of executive function, including working memory, reward sensitivity, and inhibitory control. Various hypotheses exist relating executive functions to body mass index, BMI. One prominent theory is that the role of executive function in planning and decision-making, response inhibition, and reward evaluation influences food intake, contributing to increased BMI. Functional magnetic resonance imaging studies have demonstrated that the dorsolateral prefrontal cortex is differentially activated in obesity in both adults and children. This region is critical to cognitive control over eating, as well as reward-motivated behavior via its links to the mesolimbic and mesocortical regions of the brain. Other regions of the prefrontal cortex, such as the orbital frontal cortex, have also been linked to obesity. The orbital frontal cortex is involved in inhibition and reward processing, and has been shown to be differentially activated in people classified as either lean or obese, depending on the level of satiety, including in children and adolescents. In summary, while the direction of causal association between BMI and executive function is not well understood, neuroimaging studies support the hypothesis that cortical structure and function are important to characterizing the relationship between executive function and BMI. This video will present the findings from a study that examined the association between BMI, cortical thickness, and executive function in 342 children between 9 to 11 years of age. As expected from prior literature, many demographic and biological variables were related to BMI and executive function. Males were significantly heavier than females. Birth weight was significantly associated with BMI, as was household income, race, and level of parental education. Results support statistically significant associations between BMI and executive function, between BMI and cortical thickness, and between cortical thickness and executive function. Individuals with higher BMI tend to have lower scores on executive function tests and thinner cerebral cortices, while individuals with a thinner cerebral cortex tend to have lower scores on executive function tests. The association between BMI and executive function may be mediated by their shared relationship with the thickness of a subset of regions in the prefrontal cortex. The data are consistent with a mechanism whereby prefrontal cortex changes in childhood obesity may lead to altered regulation of inhibitory control and risk-taking behavior, and further difficulties in weight control later in life.